Now, guys, now I think I am audible. I am perfectly audible now. Good afternoon, everyone. Welcome to the session. Am I audible? Am I audible now? So if it is a yes, please send a thumbs up, thumbs up, thumbs up. Quick, please send a thumbs up. Great. So welcome to the class, students. Welcome to Unacademy NEET English. I am your biology educator Ambika Sharma, and it is the part 2, right? It is the part 2 of your chapter cell. So in the first part, we have discussed the basic things related to this particular chapter. We have discussed the cell theory, about the discoveries. So let's revise it quickly and then let's move to the discussion of the prokaryotic cell. So today we are going to discuss prokaryotic cell in detail. So are you guys excited for the class? If you people are not excited for the class, then please, if you are clear about NEET 2025, right, if you have decided that you are going to prepare for the NEET 2025, then you have to be excited, you have to start your preparation again. Okay, I know the situation, it is quite chaotic, it is still chaotic, right, it is still chaotic, but still we cannot compromise our studies. Okay, so as a student, you need to start the things. As a teacher, I need to start the things, isn't it? Isn't it? Okay. So let's start the class, and before that, make sure that you hit the like button, right, bache? You subscribe our channel, and guys, you know that we are also taking our regular batches. So here in the description box, just have a look. There is a link. Click on that link, and at ₹5499 you guys can be the part of our batch, and not just our batch, you will get the access of all the batches that are running across the Unacademy platform, right, students? So proper test series, proper class where your NCERT will be discussed again and again, where your NCERT, we will try our best. We are not just trying our best, we are already making your NCERT easy and interesting, and you know that paper is going to come from that. So you guys can join, right, you people can join this particular course. And one more thing, students, yesterday I posted one video, right, about the difficulty level of the NEET, right? So here in this particular video, you will see the proper analysis, okay, right? I have considered all the factors, okay, so you guys can check it out, right? So let's get started. So once again, for your quick revision, right, let's revise this particular slide. Okay, let's revise the things from this particular slide. So you people can see here, right, you guys can see here the Anton von Leeuwenhoek, he discovered, yes bache, he discovered, right, though it is a YouTube class, but still let's revise the things and let's move to the new topic. What say people? What say people? Yes. So guys, if you are talking about the academic classes, you guys are going to get my classes somewhere in between 12 to 1 p.m. Okay, right, that will be the timing for my class, and from Monday to Thursday I'm going to take it. Okay, from Monday to Thursday you will get my classes. Fine. So now let's focus here, students. Let's have a quick revision. So Anton von Leeuwenhoek, he discovered the first living cell. We have discussed it in last class. But who coined the term cell? Who discovered the cell for the first time? Robert Hooke, right? But it was a dead cell. It was not a living cell, it was a dead cell. Okay, so Anton von Leeuwenhoek, he discovered the first living cell. Secondly, Robert Brown, he discovered the nucleus in the roots of the orchid plant. Okay, so the next thing is Matthias Schleiden in 1838. Yes, year is important. If you people will ask me, yes bache, year is important, and you guys can see here, right, Matthias Schleiden, he was a botanist, and Theodor Schwann in 1839, he was a zoologist. Okay, so who modified cell theory? Who modified cell theory, guys? Be quick. Yes. Arey, one student is saying, Ajay, right, it's second attempt, so biology is good, then why watching classes? It's very simple. If you think that your biology is good and there is no need for revision, there is no need for classes, then again you are making a mistake. So please take care of that, right? Please take care of that, okay? Right? Done. So now what is the next thing? Next thing, right? So I am asking that who modified the cell theory? Answer my question, guys. Answer my question. See, students, if you are not interested in revising, then I think it is of no use to take class on Sunday, right? I can only see unnecessarily asked questions. Satya Prakash, I am very happy that you are attending the class, but please maintain the decorum of the class. Okay, Satya? Please maintain the decorum of the class. I know. Right? You don't need it now, but you know, you have to set an example for the juniors. Yes. Who modified the cell theory? The question is, who modified the cell theory? Exactly, Rudolf Virchow. Rudolf Virchow, 1855. So actually, the cell theory given by Schleiden and Schwann was not explaining that how new cells were originating. So Rudolf Virchow, he gave us one formula, right? Omnis cellula e cellulae. He gave us one formula. Okay, omnis cellula e cellulae. It's just like a mantra that you need to chant. Okay, it is omnis cellula e cellulae, which means that cell forms from pre-existing cells. Right, cells forms from pre-existing cells. So next is, right, about the next scientist we will discuss later. Okay, so already in our NCERT we have discussed everything. We have covered the overview of cell, right? We have covered these diagrams as well, right, bache? So today the topic that we need to start is prokaryotic cell. So basically we are going to discuss about the prokaryotic cell in detail. What we need to do, guys? We need to discuss about the prokaryotic cell in detail. So are you people ready for it? Yes. Are you people ready for it? Right. So what are we going to cover, students? We are going to talk about the prokaryotic cell. Okay. I hope the brightness and everything is fine. Yes. Yes, I hope it is fine. Still, let's keep it better. Okay? Okay? Chaliye. So let's start. Okay. So the point is prokaryotic cell. So P for primitive. What this P is telling me? P is saying that it is a primitive cell. What kind of cell it is? It is a primitive cell. And this karyon word is associated with the nucleus. So bache, here this prokaryotic cell, it is a primitive cell in which we will not see, right? We will not see a well-defined nucleus. So it doesn't have well-defined nucleus. Are you getting my point? It doesn't have well-defined nucleus. Are you getting my point? Are you getting my point? Right. So this is a prokaryotic cell. Now, if I have to relate it with the evolution, right, I'm going to relate it with the evolution. And yes, students, again, I will repeat one thing. Yesterday, I was checking some YouTube videos and I have seen, right, some YouTubers, they are saying that in NEET 2024, some questions were out of NCERT, especially in biology, but it was not like that. Okay. So sometimes when your teacher, they explain few things which are, not directly not there in the NCERT, but still, if they say that yes, these things are important and they will make your NCERT easy, so please follow that. Like you remember, if you remember people, that Mesozoic era, Proterozoic era, there was one question in NEET 2024. So even if you will see my lectures on YouTube, we discuss such kind of questions because they were, right, that yes, that particular question was there in the previous year papers. Okay, so you have to understand it, right? The teachers are not here to just translate the things. They are not just here to translate the lines from English to Hindi. Of course, they are not going to do that. They are going to make your NCERT easy. And that should be your approach. I have seen that if teacher go little beyond NCERT, students, they start crying. It is of no use, guys. You try to focus here, right? You try to focus. If your teacher is saying that it is important, then consider that, okay? Okay, I know my students are good. They will listen to me. So as I said, that primitive cell, right, they don't have a well-defined nucleus. Now, I am going to relate the things a little bit with the evolution. See, when we talk about the evolution, right, you will discuss it in class 12th, right? So one theory which is chemical, which is most acceptable. So in that theory we talk about the chemical evolution. Yes, we talk about what? We talk about the chemical evolution, and in that theory we talk about the biological evolution. No need to write it down in your notes, just listen to me, I will make it easy for you. So basically they say, right, basically they say that we will have, ultimately that thing is same, that atoms, they will form molecules, will form elements, will form compounds, inorganic compounds, organic compounds, like this. Okay, inorganic compounds, organic compounds, and that organic compounds, right, monomer, polymer, finally they will form some aggregates, right, some protobions, and then when the evolution will be there. So the first kind of cell that we are going to see is prokaryotic cell, right, prokaryotic cell, right, this is what we are going to see. And from these prokaryotic cell, with the time, okay, with the time the eukaryotic cells are going to evolve. Are you getting my point? From these prokaryotic cells, with time, what is going to happen, bache? The eukaryotic cells, they are going to evolve, isn't it? Who's going to evolve? The eukaryotic cell, they are going to evolve, right? Right, so this is what you need to keep in your mind. Okay, so now when we talk about the prokaryotic cell, so we know that there is a 5 kingdom classification system. What do we know, students? We know about the 5 kingdom classification system, and as per that 5 kingdom classification system, we have kingdom Monera, right students, we have kingdom Protista, we have kingdom Fungi, then Plantae and Animalia is there. Okay, then Plantae and Animalia is there. So out of these 5 kingdoms, so it is given by this 5 kingdom classification system. Who gave it? It was given by R.H. Whittaker. Again, it is from NCERT only. Okay, it is from NCERT only. So Monera, this kingdom, it includes prokaryotes. Okay, this kingdom, it includes prokaryotes. It includes the primitive cell, the bacterial cells. Right, it includes the primitive cells, the bacterial cells. Is it clear? Right, this is the first point. Now, so bacteria, basically when we are talking about the prokaryotic cell, we need to talk about what? We need to talk about the bacteria. We need to discuss about the bacteria. Okay, so let's start. So while discussing the structure, right, see, you make sure that you note down the important points. Let me draw it here. Right, it is like this. Now, just have a look. Okay, just have a look. See, prokaryotic cell. What is it? It's a bacterial cell. It is a prokaryotic cell. So first of all, we will talk about the outer envelope. Right? We are going to talk about the outer envelope. So you know that in the last class also, students, we have discussed the overview of the cell, if you remember. Right? We have discussed the overview of the cell. And in that overview of the cell, I made it very clear that there will be, right, there will be we are not going to see a well-defined nucleus here. Plus, we are not going to see any organelle in the prokaryotes except ribosome. Right, only one organelle we are going to see here in the prokaryotic cell, and that organelle is the ribosome. Right, that organelle is the ribosome. And as I said, right, as I said that ribosome is a non-membrane bound organelle. It's a non-membrane bound organelle. Clear, bache? So basically in prokaryotic cell, only one organelle will be discussed, that is ribosome. That is a protein factory, right, and that is a non-membrane bound organelle. But ma'am, why are you telling us this thing? Because I want to make one thing clear, that when we talk about a prokaryotic cell, it is only having the outer covering. It doesn't have, right, it doesn't have the compartmentalization of that cytoplasm. It doesn't have any organelle which is having membrane, and that is doing the compartmentalization of the cytoplasm. So when we talk about a prokaryotic cell, we say that here it is having the one envelope system. right? What kind of system are we going to see here? In the prokaryotic cell, we will see one envelope system. Now ma'am, what is the meaning of one envelope system, right? So some students, they confuse it. According to them, prokaryotes are having only one outer covering. No, it simply means that only outside, right? Only the outer side is having the covering. Inside, we are not going to see anything like that. Only the outer side is having the covering, right? Within the prokaryotic cell, we are not going to see anything like this. Is that clear, Bhavna? Right, Sheikh, Ajay, Shruti, is that clear? Is that clear? Yes. Okay, so it is having one envelope system. Now, let's start the discussion of outer envelope. Right, let's start the discussion on outer envelope. Outer envelope means outer covering. Okay? Outer envelope means outer covering. So let's start from the inside. See, again, have a look. 1, 2, 3. So basically, when you talk about the outer envelope, you are going to talk about the 3 coverings. What are we going to discuss, students? We are going to talk about these 3 coverings. So innermost is your plasma membrane. Innermost is your plasma membrane. This middle one is your cell wall, and this outer one is your glycocalyx. This outer one is your glycocalyx. Please repeat it with me. Inner one, plasma membrane. Middle one, cell wall. Outermost is the yes, bache, outermost is the glycocalyx, right? Everyone note it. So these are basically the outer envelopes, right? These are the outer envelopes, or you can simply say cell envelope, means the covering of the cell, right? The covering of the cell. Is it clear? Is it clear? Yes? Tell me, is it clear? So outermost is glycocalyx, then followed by cell wall, and then the plasma membrane, right? Then the plasma membrane. Yes. So I hope you don't have any doubt here. Yes, I hope you don't have any doubt here. Okay, so now let's start the story from this plasma membrane. Okay, from where are we going to start the story? We are going to start the story from the plasma membrane, right? So bache, plasma membrane is just like the eukaryotic cell membrane, right? Here plasma membrane is not very different. It is just like the eukaryotic plasma membrane. Wait, let me explain. So plasma membrane and cell membrane is the same thing. Okay, plasma membrane and cell membrane is the same thing. So basically it is thin, right? And it interacts with the outside environment, of course. Right, it is thin and it interacts with the outside environment. It is thin and it interacts with the outside environment. One thing. Secondly, bache, it is semi-permeable in nature. What is the word? It is semi-permeable in nature. Can you explain it to me that what is the meaning of semi-permeable? Can you just explain it to me? What is the meaning of semi-permeable? Anyone in the class? My first point is that it is thin, it interacts with the outside environment, right? Then I am saying that it is semi-permeable in nature. So what is the meaning of semi-permeable? Let me mention it here. I will explain 3 words. One word is bache permeable, one word is semi-permeable, and then it is impermeable. Okay, impermeable. Then the word is impermeable. Okay, permeable means to permit, right? That is how you can relate. Permeable means to permit. Yes, bache. Permeable means to permit, means to allow, right? So it means to allow. So to allow the entry and exit of the things. Okay. So this is an outer covering. This is an outer covering. Yes or no? This is a cell. It is having an outer covering. So if it is allowing the things to come in and to go out, right? So it means it is being permeable. It is allowing the solute or the solvents to cross it, right? That is the meaning of that is the meaning of being permeable. Now the next word is impermeable. What is the meaning of impermeable? Obviously, It doesn't allow, right? It doesn't allow the entry and exit of things, right? It doesn't allow the entry and exit of things. It is being impermeable. Now, ma'am, what is the meaning of being semi-permeable? It will allow the movement of solvent across it, right? Semi-permeable membrane. It will allow the movement of solvent across it. Water can cross it. Okay, bache, water can cross it. So we also use one more word, right? Even in 9th class we have learned this. So one word is selectively permeable also. One word is selectively permeable also, or differentially permeable. Do you know that? One word is selectively permeable or differentially permeable. Do you know that? Yes. Yes. One word is selectively permeable, okay, or semi-permeable. So when you talk about the word, right, when you talk about the word selectively permeable, so when it is selectively permeable, so as per the need, it will allow the entry of solute or solvent. Here I am not specifying solvent. Here I am not specifying solvent only. I am saying as per the need, right, as per the need, the solute or solvent can cross the plasma membrane. Right, the solute and solvent can cross the plasma membrane. Are you getting my point? Yes, are you getting my point? Sure, bache? Are you getting my point? Okay, so now see, focus here. So we are saying that this is the plasma membrane. It is thin. It interacts with the outside environment, of course, and it is semi-permeable in nature. What is it, bache? It is semi-permeable in nature. So I think the meaning of all the things, right? I think you are clear with the meaning, isn't it? Isn't it? Yes. So ultimately here what will you see? You will see that phospholipids are present, because same thing we will study in the eukaryotic cell membrane. So phospholipids are present here, proteins are present here, and there are some polysaccharides that we will see, right? Right. So when you talk about the composition of the cell membrane, so definitely phospholipids are present. We know that there will be a phospholipid bilayer, proteins are there, and some amount of polysaccharides. Clear, bache? Some amount of polysaccharides. Clear, bache? Clear, bache? So I hope you know this. If you don't know it, please write down. I am saying that plasma membrane of eukaryotic cell and prokaryotic cell, its composition is somewhere same. Right, it is same. It is having same function and same way it is present. Right, so I am saying it is thin, interacts with outside environment, it is semi-permeable in nature, it is having phospholipid proteins and some amount of polysaccharides also. But bache, if you know that in the animal cell, right, in the animal cell membrane, in the case of animals, the cell membrane is having the cholesterol. Right, the cell membrane is having the cholesterol, but in bacteria, cholesterol, which is a sterol, sterols are absent. Right, you are not going to see cholesterol or any other sterol in the bacterial cell membrane. Is that clear? In the bacterial cell membrane. So basically it lacks true sterols. It lacks true sterols. Clear, bache? Right, so in bacteria, true sterols are absent. Instead of that, hapenoids are present there. Right, hapenoids are present there. Right, bache? So what is present there? They are having the hapenoids or hopenoids. Let me check the spelling. Yes, so hopenoids are present there. Okay, so cholesterol is not there. So it can be asked as a question. Fine, it can be asked as a question that true sterols are absent. Fine, true sterols are absent. So any doubt? Yes, any doubt? Tell me in the cell envelope, any doubt? Right? So what is the function of these hopenoids? They will make cell membrane stable. Okay, they are going to stabilize that cell membrane. Fine, bache, they are going to stabilize the cell membrane. Like, so it is going to stabilize the cell membrane. So is it clear? Now let's talk about the cell wall. Now let's talk about the cell wall. So anyone in the class who can tell me about the function of cell wall and the composition of cell wall? Yes, bache. Can you just tell me the composition of cell wall? Yes. Yes. Anyone? Anyone in the class? Anyone in the class? Bache, it will na, exchange is possible, semi-permeable in nature it is. Right, Bache Ronald? It is semi-permeable in nature. So that is how the exchange is possible. That is how it is interacting with the surrounding. So cell wall is going to determine the shape of the cell. Right, it is going to determine the shape of the cell. Okay. Right, what will it do? It is the cell wall. So this cell wall is going to determine the shape of the cell, and obviously it will provide the structural support. Right, without any doubt, right, it determines the shape of the cell and it provides the structural support to the bacteria. Right, and will prevent it from, and it prevents it from bursting and collapsing of bacteria. Right, it prevents the bursting and collapsing. Okay, bursting and collapsing of bacteria. Is it clear? Is it clear? This is the role of cell wall. Understood? This is the role of cell wall. So can you just give me the example of any bacteria which is without cell wall? Yes. Thank you, Bache Harsha. Thank you. So can you just tell me the example of a bacteria which is without cell wall? I think you can do it. Right? Yes. Anyone? Guys, if you don't know that, please note down these important points. Okay, do not miss it. So the answer is mycoplasma. In the last class I have explained it. So it's a bacteria without cell wall. It is a bacteria without cell wall. Okay, so the example is mycoplasma. So because it is without cell wall, right, bache? So it is pleomorphic. What is the meaning of being pleomorphic? Yes, so it is pleomorphic, means it is having different shapes. Right, what is the meaning of being pleomorphic? That it is having different shapes. Right, here the shapes are different. Okay, bache, it is having different shapes. So that is why mycoplasma, they are also known as jokers of microbiological park. Okay, they are known as jokers of microbiological park, and sometimes we also call them as jokers of plant kingdom. Now, if you are thinking that how can we say that they are the jokers of plant kingdom, it's very simple, students, right? So when they were discovered, they were placed in plant kingdom. Okay, but because of their different shape, they were basically scientists, they use this terminology for them, that they are the jokers of microbiological park or the plant kingdom. So mycoplasma is the bacteria which can survive, right, and it is without cell wall, and let me tell you, it can also survive without oxygen. Right, so yes, PYQ alert, it's a previous year question. Fine, it's a previous year question, so don't miss such points. Right, so now let's talk about the composition of the cell wall. What the cell wall is containing. So anyone in the class, can you tell me the composition here? Yes. Yes, anyone in the class? Anyone in the class? Just say when you talk about the cell wall, right? In the case of plant cell wall, we are having the cellulose, but here in the cell wall of this bacteria, we are going to talk about the peptidoglycan. right? Peptidoglycans. We also use one more word for it, that is murines. Murines, the word is peptidoglycan. Peptido is that protein part, that amino acids are present. Glycan is that carbohydrate part, right? So no need to cram anything, please understand. So peptidoglycans are going to make it. Peptidoglycans are going to make it. So what is the word that we use for it? It is NAM, murines. So here there are two things that we need to remember, and question can come from this part, NAM and NAG. So basically NAM and NAG, they are making the cell wall of the bacterial cell. So it is N-acetyl muramic acid. It is N-acetyl muramic acid, and it is N-acetyl glucosamine. It is N-acetyl muramic acid and it is N-acetyl glucosamine. So these two are present, these two are that glycans, right? These are the amino sugars basically, bache. These are the amino sugars basically, bache. Okay, so these amino sugars are alternatively linked with amino sugar chains. Okay, okay. Right, so what do we have in the cell wall? Peptidoglycans, murines, right, bache? So we are having NAM, NAG, these are the murines there, these are that amino sugars there, and acetyl muramic acid and acetyl glucosamine, right? So these are, this is that glycan or amino sugar part. Okay, so these chains, right, these chains are alternatively, they are cross-linked with small peptide chain. They are cross-linked. Okay, they are cross-linked with small peptide chain. Okay, any doubt? Yes, so question can come from this part. Yes, bache, question can come from this part, right? So I said cross-linked with small peptide chain. Now I will answer one very important question here. Okay, so students, you know that we have the antibiotics, right? You know that we have the antibiotics, isn't it? Antibiotics. Anti means against, bio means life. So it is something which is against life. So here we say, right, here we say that these antibiotics, they are going to destroy the bacteria. If you know it, then do let me know in the chat section, quick. If you know it, do let me know in the chat section, quick. Yes. Yes, yes, yes. Antibiotics. So we have, I'll give you the example of two antibiotics. One is penicillin and another is lysosome. Actually, these antibiotics, they also behave in a different way. Penicillin, it is the first antibiotic discovered. So this penicillin, it prevents cross-linking of these peptides, right? Just say, let's say these antibiotics, they are going to kill bacteria. What these antibiotics are going to do? They are going to kill bacteria. They are going to destroy it. And there are different ways. Just let's suppose if you have to, if someone is going to kill someone, then obviously there are different ways. There are different ways. So same is the case here. When we are talking about the antibiotics, there are different ways by which they can kill the bacteria. For an example, if I talk about the penicillin, it will not allow that cell wall, right? It will not allow that cell wall to form that cross-linking with that peptides. Means it will not let the synthesis of that cell wall properly. Okay, how this penicillin is going to kill the bacteria? It will not allow the cross-linking of the peptides. It will not allow, right, that bacteria to form these, right, the cell wall properly, right? It can prevent cross-linking. Okay? Okay? Yes or no? And lysozyme, how that lysozyme is going to act? You know the lysozyme, it is present in saliva, right? It is present in tears. So how this lysozyme is going to kill bacteria? It can hydrolyze, right? It can hydrolyze peptidoglycans. It can hydrolyze it, right? So one will not let the cell wall to synthesize properly and another will just destroy it, right? Will just hydrolyze it. Clear? Clear? So this is what we need to know. Understood? This is what we need to know. Now, bache, in the cell wall, on the basis of cell wall, we have two types of bacteria. We have two types of bacteria. One is gram negative bacteria and another is gram positive bacteria. On the basis of this cell wall only, gram negative bacteria and gram positive bacteria. Do you know about it? Yes, I'm saying that on the basis of cell wall, we have two types of bacteria. One is gram negative and another is gram positive. One is gram negative and another is gram positive. So can you tell me about it? Can you tell me about it? Can you tell me about it? Gram negative or positive, whatever differences you know. Yes, whatever you know related to this. Huh? Students, it's very simple. Here we discuss about a scientist. Okay, the name of the scientist is Christian Graham. The name of the scientist is Christian Graham. He made one stain. Stain means that color, right? Like if we have to see something under the microscope, if we will color it with different colors, right? Like let's say if you're talking about the cell organelle, if nucleus is having different color, right? If other organelles are having different color, so under the microscope we can see it clearly, right? We can see it clearly. So when you talk about the Christian Graham, he made one stain, that kind of color, right? So bacterias that gives color with Gram stain are considered as gram positive. Gram positive means you can color such bacteria, right? You can color such bacteria with the help of this particular stain, right? So bacteria that do not give color with Gram stain, they are gram negative. Okay, they are gram negative. So in this way we differentiate bacteria positive and negative. But the point here is, ma'am, right? But the point here is why some bacteria can give color with Gram stain and why some bacteria they cannot give color with Gram stain. What is the point here? What is the thing? Why is it so? But it is on the basis of cell wall. Let me explain this. So there is a trick. Come in and stain. Right, there is one trick, that is come in and stain. Do you know about it? Yes, do you know about it? Quick, right? I'm going to read your charts. Quick, answer it to me. Do you know about it? Come in and stain. This is the trick. Come in and stain. Come in and stain. That is a trick basically. Come in and stain. So obviously for that, first of all, what do we need to do, right? What do we need to do? First of all, we have to collect the sample of bacteria, right? So basically, I don't know whether you have done it in your school or not. First of all, you are going to collect the sample of the bacteria, right? Ultimately, this is what you know. And there are some steps of the staining that you need to follow. So I will not discuss that steps in detail, but whatever is important for your exams, let's discuss it. Just say there will be the trays in which you have to put your sample, that is your bacteria. Then you have to add the color. So come in and stain for C. C means crystal violet. That is the main stain. That is the primary stain. Right? That is the primary stain. That is the main stain. Right? Crystal violet. So first of all, you have to add this particular stain. Then come in I. I means iodine. I means iodine. Iodine is going to fix that stain. Like let's say you want to paste a painting. Right, you want to put a painting, okay, so you can use the Fevicol to stick it. In that way also, you can use the double tape to stick it. So same way, we are going to use the primary stain first, right? We are going to use the primary stain first, and then after that primary stain, after that primary stain, what we have to do? We need to put a mordant. What we need to do, we need to put a mordant. What we need to do, everyone in the class? We have to add a mordant, and that is grams iodine, right? That is grams iodine. So you are, this is the word mordant. Iodine is a mordant. It is going to fix the stain. What will it do? It is going to fix the stain. I hope you guys can visualize that you are going to take a sample where bacteria, you are in a tray, you are going to take the samples, then you are going to add the crystal violet stain, which is the main stain, primary stain. Then after that you will add the grams iodine, right? Bache, that is basically the mordant that is going to fix the stain. And then after that this A, right? What is the meaning of this A? A means alcohol or acetone. So it is for washing. It is for washing. So after it you are going to add either alcohol or acetone that will wash the color. It is for the washing. So after the washing, right, let's say you have a bacterial sample. Okay students, let's say you have a bacterial sample, right? So here in this bacterial sample you have added the crystal violet, iodine, and then you wash the sample with the acetone or alcohol. So you will see that some bacteria, right, that stain will come out from it. The stain will come out from it. Okay, bache? So after this, basically this is the procedure, right? This is what you have to follow. You have to add, as for secondary stain, that is your safranin. That is your safranin. It is the secondary stain. Okay. So now what's going to happen? What is going to happen? The bacteria which are gram positive, even after the washing with alcohol and acetone, they will retain the color of this particular stain. But in the case of gram-negative bacteria, after this washing with alcohol or acetone, they will just lose that gram stain, and then that is why we add one more stain, so that these gram-negative bacteria, they can pick up this safranin, so that we can see them. Okay, so that we can see them. So why is it happening? As I said, it depends upon what? It depends upon this, right? The structure of cell wall, right? The structure of the cell wall. So it is a gram positive bacteria, it is a gram negative. So when you see a gram positive bacteria, please have a look. In the case of a gram positive bacteria, we have a cell membrane. Then outside this cell membrane, bache, there will be one but very thick cell wall. Okay, this is your gram positive bacteria, right? This is your gram positive bacteria. So in your gram positive bacteria, outside this, what is it? It is your cell membrane. It is your cell membrane. And what is it? It is your cell wall. So cell wall is made up of peptidoglycans. So it is very thick, if you guys can see. Yes, if you people can see, it is very thick, right? It is very thick. So in that particular step, I am telling you now, the washing will be done. I am telling you now, the washing will be done. The alcohol or acetone will be added. So when alcohol or acetone will be added, right, so that alcohol and acetone cannot create the pores here in the cell wall, and that stain cannot go out, right? So that is why gram positive bacteria, because of this thick cell wall, it is going to retain the color of that, what do we call it? Of that gram stain. But now when you talk about the gram negative bacteria, so here it will be like this. It will be like this, right? Now ma'am, what is it? Simple. This is the cell membrane. Okay, if you will see, the cell wall is not so thick. Plus here in gram negative bacteria, you will see one more outer envelope. You people are going to see one more outer envelope, and that outer envelope is again made up of lipids. So when you add alcohol or the acetone, it is going to create the pores here. And that stain will come out. Okay, bache, that stain will come out. Clear, bache? So because of this, you can see that some bacteria, they are colored with gram positive, they are colored with gram stain, and some bacteria, they are not. Can you see it? Yes students, can you see it? So in your gram positive bacteria, you see that teichoic acid, right, is present in the cell wall, right? This teichoic acid is present in the cell wall. So have a look, gram positive and gram negative. Okay, so this is a cell membrane, this is a cell wall, the space in between is the periplasmic space. Okay, it is the periplasmic space. So peptidoglycan, this is the outer membrane, again it is made up of lipids. So these are, right, liposaccharides. So basically when you will add alcohol or acetone, you will see that the pores will be created. Okay, what are you going to see? That the pores will be created. So on the basis of that, we can see the color as well. After adding the stain, we can also study the shape of the bacteria. Okay, we can also study the shape of the bacteria. So tell me if there is any doubt. If you have any doubt, do let me know. If you have any doubt, do let me know. No doubt? Okay, so you can write down the examples also. Right, you can write down the examples also. Okay, so now you know the reason. So gram negative bacteria mein Escherichia coli. Bache, you have to check the spellings. It is E. coli, na? Escherichia coli it is. Okay, you have Neisseria gonorrhoeae. That is also the example of gram negative bacteria. It causes the sexually transmitted infection. So here you have the example of Staphylococcus species. You have the example of Streptococcus species. You have the example of Clostridium. So these are few examples, and we are going to use these names in our biology. Okay, bache? Okay, done? Quick. Quick. Yes? Clear? Is it clear? Is it clear? I hope it is. Right? I hope it is. So that is what you need to remember. Okay, that is what you need to remember. Now the next thing is Glycocalyx. This Glycocalyx is also known as cell coat. Okay, this Glycocalyx, it is also known as cell coat. It is the outermost covering, and we will see that it is of two types. Okay, one is your slime layer. One is your slime layer and another one is the capsule. Another one is the capsule. So Glycocalyx, what is it, bache? This Glycocalyx is the outermost covering, students. It is the outermost covering, right, bache? So it differs in composition and thickness among different bacteria. So if it is, so it can have two types, the slime layer and the capsule. The slime layer and the capsule. Clear, bache? It is slime layer and the capsule. So slime layer is nothing but it is a loose sheath. It is a loose sheath, slimy, right? Kind of jelly-like. It's a loose sheath, right? It's a loose sheath and it is thick and tough. It is thick and tough. So some bacteria, they can have this slime layer, and some bacteria, they can have this thick and tough capsule. So as I said, in different bacteria, the composition of this Glycocalyx, it varies, plus the thickness of this Glycocalyx, it varies. Clear, bache? Clear? So it is the outermost mucilaginous sheath. It is the outermost mucilaginous sheath, right? So mucilaginous, which can hold water also. Okay, it is the outermost mucilaginous sheath, bache. So it is having the mucopolysaccharides, right? It is having the mucopolysaccharides. These are modified polysaccharides with or without proteins. So overall, this is the Glycocalyx, the outermost covering, right, having mucopolysaccharides, and it can be with or without proteins. Done, bache? With or without protein, right? So if I talk about their function, so remember, slime layer will protect cell from loss of water and nutrients, right? From loss of water and nutrients. That is how it is going to protect it. okay, from loss of water and nutrients. And when you talk about the capsule, it protects it against desiccation, against antibiotics. Understood? So that is the role of this. That is the role of this glycocalyx, right? And even this capsule also protects from viral attack. Tell me, any doubt? So this is about the glycocalyx. This is the outermost covering. It is about the glycocalyx. It is the outermost covering. Okay. So it is having the two types. Understood? It is having the two types. Understood? Yes, bache? Yes? Okay. So now let's talk about the next thing, that is the cytoplasm. But before that, let's revise this portion from NCERT. So if you guys are having any doubt, do let me know. If you people are having any doubt, do let me know. Yes? Any doubt? So, prokaryotic cells, they are represented by bacteria, by blue-green algae, by mycoplasma and PPLO. Okay, very important information again. Right? So, it is represented by bacteria, right? Blue-green algae means, what is the meaning of blue-green algae? It means photosynthetic bacteria. It means photosynthetic bacteria. Okay, so the next part is mycoplasma. I already told you it is the bacteria which is without cell wall. So you are not going to get such lectures where teacher is providing his or her notes plus they are also making your NCERT easy. So be consistent, follow this channel only. That is going to help you in your NEET preparation, right students? So it is without cell wall, this mycoplasma, and you know that it is the smallest living organisms, right, which is 0.3 micrometer. Then pleuropneumonia like organism, it is just like a mycoplasma and it is 0.1 micrometer. Okay, okay, so they are smaller. These bacterial cells, they are smaller. They multiply very quickly in comparison to the eukaryotic cell. And as I said, they have different shapes and size. So one question can come from this shape part also, right? It is again a previous year question. Okay, so if it is spherical, right, you will use the word coccus, like streptococcus, plural is cocci, right? If it is a rod shaped, means if it is a cylindrical, we will use the word bacillus. Bacillus is the word. If it is comma shaped, something like this, right, then the term that we are going to use here is vibrio, right, comma shaped. And it can be spiral also, right, spiralum. What is it? It is spiralum. Understood? Okay, so you can see this. So it is bacillus, rod like, coccus, spherical, vibrio, comma shaped, and spiralum, spiral, right? It is also important, right? Okay, so see, this diagram is important. Whatever is given in NCERT, it is important. We cannot miss that. So you guys can see a typical eukaryotic cell, its size is 10 to 20 micrometer. Right, its size is 10 to 20 micrometer. A typical bacteria, its size is 1 to 2 micrometer. PPLO 0.1 micrometer and viruses 0.02 to 0.2 micrometer. Understood? To 0.2 micrometer. Clear, bache? Right? So prokaryotes, they have a cell wall and you know that it is absent in mycoplasma. Right? So fluid matrix which is filling the cell is the cytoplasm. So now we have to discuss the next part. So any doubt here? Your NCERT is easy? Your NCERT is easy now? Now your NCERT is easy? Okay. So before going ahead, some students are again and again typing it in the chat section because bache, why can't you just let me complete it in my own pace? So students, when you talk about the bacteria now, this is what we are going to study in kingdom Monera. Okay. So in kingdom Monera, yes, bache, in kingdom Monera, there are two things, right? You are going to talk about the true bacteria that is your eubacteria, and one is the primitive bacteria, that is known as archaebacteria. So in the case of archaebacteria, cell membrane and cell wall structure is different. Okay, cell membrane and cell wall structure is different. So now you have to, that is your homework, you have to write the differences in these two. Fine, you have to write the differences in these two. Okay, right. So eubacteria and archaebacteria, how their cell wall and cell membrane is different. Right, how their cell wall and cell membrane is different. This is what you need to tell me. So next is the cytoplasm. So cytoplasm, you know that, as we said, it's a fluid matrix. Okay, what is that cytoplasm? It is that fluid matrix, isn't it? It is that fluid matrix. Colloidal it is. There is no compartmentalization because it doesn't contain the organelle, and the organelle which is present in bacteria is ribosome, which is again a non-membrane bound organelle. Clear bache? Clear bache? So this cytoplasm is having various structures, like cell membrane is going to form some finger-like extensions. You know that? Cell membrane is going to form some finger-like extensions. We call it as mesosome. And where is that mesosome present? Obviously in the cytoplasm. So there are various structures which are present in cytoplasm. So it is a mesosome, which is a special membranous structure, right? This mesosome is what? It is a special membranous structure. Okay, so it is basically the infolding of the cell membrane, the finger-like infolding of the cell membrane. It is the extension of the cell membrane. Is it clear? Right, bache? It is the extension of the cell membrane. It is extension of the cell membrane, and in the form of vesicles, means that extensions can have different shapes, like vesicles, tubules or lamella, right? They can have different shapes, right? So what are we talking? We are talking about the mesosome. We are talking about the mesosome. Clear? So it's a special membranous structure. It is basically the extension of cell membrane in the form of vesicle, tubules and lamella. Now you know that in the eukaryotic cell we have the mitochondria. That mitochondria is having the respiratory enzymes. That mitochondria is having the respiratory enzymes. Right, bache? Right, bache? But in bacteria we don't have any structure like this. So somewhere this mesosome is going to help there. So this mesosome, these mesosomes, they are going to have the respiratory enzymes. Right, they are going to have the respiratory, they are having the respiratory enzymes, and obviously that will help in the cell respiration. So that is how this mesosome is going to help. Right, students, that is how this mesosome is going to help. So other than that, they will help in the cell wall formation. Other than that, it is going to help in the cell wall formation, DNA replication. You should know about the roles of your functions of this mesosome. So it will help in the cell wall formation, students. It will help in the DNA replication, right, and distribution of that DNA to daughter cells, right, and the distribution of that DNA to the daughter cells. And you know that, as I said, it contains respiratory enzymes, right? It contains respiratory enzymes. And students, do you know why? Why is it having that finger-like extensions? Why does it contain the finger-like extensions, right? Why is it containing that finger-like extension? Why? Because these finger-like extension, they will do what? They will increase surface area. So if they are because they are increasing surface area, so definitely the enzymatic content will be more, right? The enzymatic content here will be more. Understood? Understood? Yes, bache. Understood? Okay. Now in some other bacteria like prokaryotes, right? In some bacterias, right? Sorry, not prokaryotes, some bacterias, there are some other membranous extensions, right? some other membranous extension, and that will contain the pigment. Okay, so in some prokaryotes, जैसे in some bacteria like cyanobacteria, right, there are some other membranous extension. We are going to call it as chromatophores. Okay, so where are you going to see chromatophores? See, in some cyanobacteria. What type of bacteria is a cyanobacteria? Your blue-green algae, your photosynthetic bacteria, right? Your blue-green algae, your photosynthetic bacteria. Okay, cyanobacteria, your blue-green algae, your photosynthetic bacteria. Clear, bache? Right, so it is having these membranous extensions. Right, we are calling it as chromatophore. So chroma means color. So these chromatophores, they contain pigments, and you know that because this bacteria is photosynthetic, it needs that pigment for the photosynthesis, right? It needs that pigment for the photosynthesis. Understood? Okay. See, in the NCERT itself, they have given this. Right, they have given it. So let's revise it from NCERT and then let's discuss it further. So see, most prokaryotic cell, particularly the bacterial cells, they have chemically complex cell envelope. Any doubt in the first line? Please open your NCERT and answer it to me. Kindly open your NCERT and answer it to me. And answer it to me. Yes. So cell envelope is having tightly bound. It is having tightly bound three-layered structures. So outermost glycocalyx is there. Then bache cell wall is there. Then plasma membrane is there. Right. So each layer here is going to perform different functions, but they all will act together like a single protective unit. Right. They all are going to act together like a single protective unit. Right, and you know that on the basis of differences in these cell envelope, we have gram-negative and gram-positive bacteria. This is what we have discussed, bache. Right, you know about it in detail. Secondly, glycocalyx also, it differs in composition and thickness. So if it is a loose sheath, it is slime layer. If it is a thick and tough, it is capsule. So next to it is cell wall, which will determine the shape, and I told you their function also. Right, bache? Right, so here they have also explained the mesosome, right, and we have already covered that in the form of vesicles, in the form of some tubules, in the form of some coverings or layers, right. Mesosomes will form, so they will help in cell wall formation, DNA replication, distribution to daughter cells, so they will also help in respiration and secretion processes. Understood? So in cyanobacteria, what will you see, bache? You will see that there are chromatophores. What are you going to see? You are going to see the chromatophores. Clear, bache? Is it clear? Yes. 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 Done? Okay, so the next thing that we need to understand is bacteria. Okay, the next thing that we need to understand is bacteria. So just about is flagella. So see this diagram, isn't it beautiful? So here in this diagram, nucleoid, the cytoplasm, ribosome, look at these structures. We are calling it as fimbriae. This long structure, it is the flagella, and that is what we need to discuss now. Okay, that is something that we need to discuss now. So see, your bacteria, it can be motile or non-motile. It can be motile or non-motile. What is the meaning of motile? Right? What is the meaning of motile? It means movable. It means movable. Right? It means movable. Non-motile. Right? Non-motile, the bacteria that cannot move, right? It is movable, it is immovable, it is immovable, right? So if a bacteria is movable, right, how that bacteria is going to move? In the case of bacteria, you are going to see a structure, a long thread-like structure, and we call it as flagella, right? We call it as flagella. What is it? It is a flagella, and it is a very simple flagella, students. It is a very simple flagella, and it is made up of flagellin protein. It is made up of what? It is made up of flagellin protein. So it's a simple flagella, it is made up of flagellin protein. Now, why am I specifying it? Let me tell you, because I think it is the most commonly made mistake. So, in eukaryotes, okay, in eukaryotes, flagella is made up of tubulin protein. Okay, flagella is made up of microtubules. We will discuss it. It is made up of what? It is made up of microtubules, means tubulin protein. But in the case of your prokaryotes, it is made up of what? It is made up of this flagellin protein, right? It is made up of the flagellin protein. Understood? Understood? Yes? Clear? So it is the flagella. So this is composed of 3 parts. So it is having, so now bacterial flagella is having 3 parts. It's a very simple structure, you should know about it. So one is the basal body, another part is the hook, and then it is the filament. One is basal body, another is hook, and the filament. These are the 3 parts of the flagella. So see, basal body, from where this flagella will arise, then there will be a hook-like structure, and then there will be a thread-like filament, right? Then there will be a thread-like filament. Clear, bache? Clear. So filament is the longest portion, right? It is the filament that is the longest portion of the flagella, okay, of the flagella, and obviously we can see that it is projecting outside, है ना? It is projecting outside. Is that clear? It is projecting outside. Can you see that? Can you see that? Yes, can you see that? Yes, students, can you see that? So if there is any doubt, do let me know. Do let me know. So filament is the longest portion. It is extending from the cell surface to the outside. So these are the thread-like appendages, bache, right? So mainly you will see, okay, so it will show some rotational type of movement. This flagella is going to show some rotational type of movements. So it is not required for NEET, it is just extra information, rotational type of movement. So this is about the flagella. This is about the flagella. Clear, bache? So next to flagella is ribosome. Let's talk about the ribosome. Okay. Let's talk about the ribosome. But before ribosome, you tell me, do you have any doubt in the chromatophores or something? Naina? So let's talk about the ribosomes. Palade discovered it. Palade discovered the ribosome. Ribosomes are basically the protein factory. What is the meaning of protein factory? It means that they help in the synthesis of protein. Okay, and this ribosome, it is a non-membrane bound organelle. It is not covered with, right? It's a non-membrane bound organelle because it is not covered with any membrane. It is not covered with any membrane. And here in the ribosome, you will see 2 subunits, the larger subunit and the smaller subunit. But we will see the same thing in the eukaryotes also, right? But here I will discuss about the prokaryotes only. Okay, so smaller and the larger subunit is there. So smaller subunit, it is 50s, larger subunit 50s, smaller is 30s. So when you talk about the ribosome in the case of prokaryotes, It is the ribosome. It is a 70S ribosome. It is a 70S ribosome. So it is having the larger subunit, it is having the smaller subunits. Right, bache? Both the subunits, they will join at the time of protein synthesis. They join at the time of protein synthesis in the presence of magnesium ion. So it is the magnesium ion which is going to join it. It is the magnesium ion which is going to join it. Just let's say कि when relatives they visit our place, right, the siblings, they don't have the common opinion, right, they fight with each other, but mother is like, if in front of siblings, if in front of relatives you are going to fight, I'm going to beat you. So you have to behave like this, that you are together as a unit, you don't fight in front of your relatives. Same is the case here. Larger subunit, smaller subunit, they will stay separated, but at the time of protein synthesis, M for mummy, M for magnesium ion is going to join it, right, it is going to join it, and you will have the larger subunit. Okay, so you will have this particular structure. So when we talk about the 70S, what is this S? This S means Swedberg unit, right? Swedberg unit. What is this S? It is Swedberg unit. Again, it is also a previous year question, students, right? It is also a previous year question. So what is it? It is a Swedberg unit. So actually it is a sedimentation coefficient. What is it? It is a sedimentation coefficient. What's that? It is a sedimentation coefficient. Now see, I told you that there is a technique, cell fractionation, that we use to study a cell, right? Let's say in an ultracentrifuge, we will just divide this, break the cell into the components, and then we study them, right? As per their weight, they will settle down. Now, same is the case here. So as per the weight, these ribosomes, they are going to settle down. So 70S is there, 80S is there, that we study in the eukaryotic cytoplasm. But in prokaryotic cytoplasm, it is 70S. So this S is Swedberg unit. It is basically the sedimentation coefficient. So you have 50S and 30S. No need to add it. Ki 50 plus 30 is 80. How can you say 70? 9. Right, these are the name of the subunits. These are basically as per their sedimentation. 50S, 30S, 50S, 30S. So now, when we talk about these ribosomes, they are made up of ribosomal RNA, and there are some proteins which are going to make it. Right, they are made up of ribosomal RNA and they are made up of some proteins. So again, it is your homework. You will tell me that which ribosomal RNAs are making this, right, larger and smaller subunit. It is your homework, right? It is your homework. Understood? Understood? It is your homework. So ribosomes, they are the protein factories because they are the site of protein synthesis. Clear, bache? Clear, clear, clear? They are the site of protein synthesis. Understood? They are the site of protein synthesis. Now, these ribosomes, see, these ribosomes, they are also attached to a mRNA. So many ribosomes, they are attached to mRNA. So many ribosomes, they are attached to what? Attached to mRNA. So when so many ribosomes, they are attached to the mRNA, then they form a structure, and that structure is known as polysome or polyribosome. Polysome or polyribosome. So where are you going to see polysomes and polyribosomes? You are going to see it in the case of prokaryotes. Where are you going to see it, bache? You are going to see it in the case of prokaryotes. So to the single mRNA, right, so many ribosomes are attached, and simultaneously transcription, translation will occur, right? Simultaneously in the prokaryotes, you will see the transcription, translation occur. I know if you are in class 11th or in 12th, if you don't know anything from molecular basis, you cannot understand these terms. So just wait for that particular chapter, molecular basis of inheritance, then you can understand it in a better way. So transcription means when from DNA mRNA forms, and translation means from mRNA to the protein formation is basically the translation. So in prokaryotes, both the processes, they simultaneously occur. So this is the mRNA, ribosomes are attached, they will start making the protein. So this structure is polysome or polyribosome. This structure is polysome or polyribosome. Clear, bache? Clear, clear, clear? Understood? Right? So they will translate the mRNA into protein. Understood? So students, in the prokaryotes itself, there are some ribosomes which are present in cytoplasm. They are and some ribosomes, they are basically present on this plasma membrane. So there are fixed ribosomes and free ribosomes as well, right? So we have fixed ribosome and free ribosomes as well. Clear? Clear? So next thing that we need to understand is nucleoid. Nucleoid. So can you tell me what is nucleoid? Yes, can you just tell me what is nucleoid? Which new words? Ronald, which new words? If you are talking about transcription and translation, just wait for the chapter genetics. So now tell me about the nucleoid. What do you know about nucleoid? Let's discuss. What do you know about nucleoid? Guys, be quick. We are about to finish this prokaryotic cell part in next 15 minutes. So answer it to me. What is nucleoid? What is nucleoid? Exactly, है ना सृष्टि? It is the naked DNA. So basically we know that in the prokaryotic cell, we don't have a well-defined nucleus, right? So here the genetic material, it is naked. It is naked means not covered with histone proteins. Histone proteins are not going to cover this DNA. Okay, and there is no well-defined nucleus means no nuclear membrane is there, right? No well-defined nucleus, no well-defined nucleus, right? So it is a naked means it is not covered with histone protein. So we also write it. So it is basically, this structure is known as nucleoid because it is devoid of histone proteins. It is not covered with histone proteins. It is directly there in the cytoplasm and it is not covered with any nuclear membrane. Okay, it is not covered with any nuclear membrane. And what kind of DNA is present here? It is the circular DNA. It is the circular DNA. Now imagine it. Right, you are talking about a bacteria. It's a circular DNA. What is it? It's a circular DNA. Right, you can imagine a thread. Right, a thread which is forming a circle. Right, something like this. The circular DNA, the nucleoid. Right, something like this. So basically in bacteria, we also call it as genophore. The structure is also named as genophore, or you can call it as prochromosome. So in the bacteria, in the prokaryotic cell, there is only one chromosome because this is a circular DNA, right? Not linear DNA like eukaryotic cell. So it is a circular DNA. So you are also calling it as nucleoid. You also use the word genophore and the prochromosome for it, right? And the prochromosome for it. Is it clear? Is it clear? Now in the bacteria, you are also going to see then one extra ringlet, right? One extra ringlet of DNA. In the bacteria, we also see the extra ringlet of DNA. Just see. Can you see this diagram? Can you see this DNA? Can you see this part? It is your plasmid. It is your plasmid. It is your plasmid. See, this is your plasmid. Plasmid. That is something we are going to see in bacteria. So again, you can see that it is a circular DNA. Can you see that? It is a circular DNA. So we also call it as extra chromosomal DNA. Right? It is not just the chromosomal DNA in bacteria. It is also having extra chromosomal DNA, right? Which can replicate by its own. Okay? Which can replicate by its own. Okay? So basically it is not dependent on chromosomal DNA. For its replication. And why is it so? Because it is having its own, because it is having its own origin of replication. Because it is having its own origin of replication. Understood? Because it is having its own origin of replication. So what am I saying? I am talking about the extra chromosomal DNA, that is plasmid. It is present in the bacteria, right? So you will see that this plasmid is not dependent on the chromosomal DNA for its replication, means for multiplication. It can divide by its own. It can divide by its own, and then now it is a DNA. Obviously, it is having some genes, right? And that genes are going to give special property to that bacteria. Let me explain. Let's say there is one bacteria, and in that particular bacteria we can see that this plasmid is having antibiotic resistant gene. Okay, antibiotic resistant gene. Basically this bacteria is not this plasmid is having antibiotic A resistant gene, means if I will give antibiotic A to this particular bacteria, it will survive. Why? Because plasmid is having a gene which is against, right, which is having the resistance against this antibiotic. Are you getting it? So this plasmid, it is having certain genes which will give this bacteria the special properties. So you people will understand the role of this plasmid in the chapter biotechnology of your class 12th. Right, you will understand the role, the function, everything, right? And once we will discuss genetics, you will get to know what is the importance of origin of replication, what is origin of replication. We will discuss it there. Okay, we will discuss it there. So this is about the plasmid. This is about the plasmid. Now bache, here, right, ribosome is also clear. Here in this bacteria, you will see certain, of course, non-membrane bound bodies. We'll call it inclusion bodies. What are we going to call it? We are going to call it inclusion bodies, right? Inclusion bodies. Again, that inclusion bodies are also unique to this bacteria. Okay, they are unique to this bacteria. They are basically the kind of storage granules, right? So in bacteria, what are we going to see in the cytoplasm? Inclusion bodies that are basically the storage granules, जैसे glycogen. Right, जैसे glycogen granules. So basically inclusion bodies again non-membrane bound. It is the reserve food material. Okay, what is it? It is basically the reserve food material. Okay, it is basically the reserve food material in the case of bacteria. So now let's cover it from NCERT. Right, that cover it from NCERT. So here you people can see that in prokaryotes, ribosomes are associated with the plasma membrane of the cell. Yes, bache. So some ribosomes, they are associated with plasma membrane, they will be fixed, and some are in cytoplasm, and we will call them as free ribosome, right? So basically they are about 15 nanometer to 20 nanometer in size. Yes, sometimes they also ask the size. Okay, they can also ask you the size. So 15 nanometer to 20 nanometer in size, and it is made up of two subunits, 50 and 30, right? So which will be present together and form 70S ribosome. So they are the site of protein synthesis. So many ribosomes, when they attach to a single mRNA, it's polysome or polyribosome. It is discussed. Now inclusion bodies, as I said, they are the reserve food material in prokaryotes. What are they? They are the reserve food material in prokaryotic cells, right? They are stored in cytoplasm. Okay, so it is basically the storage granules. They are not covered with any membrane system. So question on the basis of inclusion body was there in 2020. It was there in 2022 as well. But yeah, for sure it was there in 2020, right? It's a 2020 ka PYQ. So it is not covered with any membrane. It is not covered with any membrane. As I said that your prokaryote is a one envelope system. It is just having the outer covering. It doesn't have any inner covering. ठीक है बच्चे? Right? So phosphate granules, synophysian granules, glycogen granules. These are the different types of granules. And in certain, right, in some bacteria like your green photosynthetic bacteria and in your blue, green and purple and green photosynthetic bacteria, gas vacuoles are also there. Right? So in the case of some bacteria, gas vacuoles are there, and obviously that vacuoles are going to provide the bio-NC. Okay, they are going to provide the bio-NC. Done, bache? Right? So they will help that bacteria to float. They will help that bacteria to float. Is that clear? So these are the inclusion bodies. Now we need to discuss two more things in the case of a bacteria. first thing is fimbriae. In that diagram, did you notice that some bristle-like structures coming out? Did you notice that? Did you notice that some bristle-like structures, they are coming out from this bacteria, from that bacteria? Yes, but we call it as fimbriae, right? We call it as fimbriae, and they are non-motile. They will not help in movement, right? It is also previous year question. They are not going to help in movement, but these fimbriae, they will help the bacteria to get attached to substratum. Substratum means surface, right? Substratum means surface. So it will help the bacteria to get attached to the substratum. Now let me explain it to you. See, I'll give you the example of one bacteria, that is your Neisseria gonorrhoeae, right? I will give you the example of a bacteria that is Neisseria gonorrhoeae. So students, this particular bacteria, it causes STI. What is STI? It causes sexually transmitted infection. It causes sexually transmitted infection. Okay, and the name of that infection is gonorrhea. Okay, it is a sexually transmitted infection. Now this particular bacteria, it is having the fimbriae, and with the help of that fimbriae, this bacteria is going to attach to the cells of the host, right? Just we are the host. So in our case, with the help of this fimbriae, this Neisseria gonorrhoeae will get attached, and then obviously will enter in our body and will cause the infection. So fimbriae, what are they? They are non-motile. Non-motile. It is not movable. It is non-motile. Clear? It is non-motile. Clear, bache? Right? So one more structure is there. And that is pili. Pili. It is made up of pilin protein. It's a kind of, again, non-motile but tubular structure. Non-motile but tubular structure. What kind of structure it is? It is a non-motile but tubular. I'll give you one example. Let's say this is your one bacteria. This is your another bacteria. This bacteria can have a tube-like structure which will help this bacteria to get attached to this particular bacteria. Okay, and we call it as sex pilus. Okay, so now with the help of this tube-like structure, this particular bacteria can pass its DNA to this. It can pass the DNA to this. So it is basically the conjugation in bacteria that I have just discussed, bache. So in biological classification chapter also we will talk about it, right? In the chapter biological classification we will talk about it. So it's a tube-like structure which will help the one bacterial cell to pass its genetic material to another, right? So it's a, I won't say that bacteria is having true sexual reproduction, but kind of sexual reproduction is there, right? So just say this is one bacteria. Okay, it is one bacteria. This bacteria is having F plasmid. It is possible due to the plasmid only. You cannot say that all the bacteria are going to do this. No, F plasmid. F means fertility. Okay, so it is F positive plasmid, but this particular bacteria doesn't have, so we will call it F negative plasmid. So this particular bacteria will form this sex pilus and will pass, right, and will pass this genetic material from this bacteria to this bacteria, and in this bacteria also we will see that F plasmid will form. So this is conjugation. So that is how this tube-like structure is going to help. Okay, so again it is also non-motile. You can read it from this part. So in addition to flagella, pilae and fimbriae, they are also, right, they are also the surface structure of the bacteria, means they are present on the surface of bacteria, but they do not play a role in motility. So for the movement, flagella will play the role, but not pilae and fimbriae. One point. Now pilae are elongated structure made up of special protein. You know the name of the protein is pilin. So fimbriae are small bristle like fibers which are sprouting out of the cell, which are sprouting out from the cell. Okay, bache. So in some bacteria, they are known to help to attach the bacteria to rocks in stream and also to the host tissue. So this is all about the prokaryotes. So in the next class, we are going to talk about the eukaryotes. But before that, answer a few questions of mine, and then I will share the PDF in the Telegram group. So guys, be consistent to this, to these classes, right? We have started this special series, the Infinity series, for the students, those who are not the part of our batches and they are just following us on YouTube. But still, I will suggest you people to join the course, bache. And for joining the course, you can see any video on our channel, right? You can see any video on our channel, and in the description box, you guys are going to get this link. So you have to click on this link, you have to add your detail, and in just 5499 you can get 1 year plus subscription. And if you want to buy it for 20 months, 24 months rather, for 2 years, so of course, right, the amount will be near about 10,000. Okay. So by joining our batch, you will have the access of all the batches on the Unacademy platform. You'll be the part of our personal mentorship group. You will get the notes as well, right? If you will buy plus subscription, you guys will get PDF notes. If you will go for iconic subscription, you will get the hard copy of the material. Plus, right till your NEET examination, you will have the access of the batches. You can join our crash course. You will be the part of test series, and you have to pay nothing. So there are no hidden charges, but that, right, no hidden charges are there. And in this one-time payment, you can access everything on our platform, right, bache? So the panel is very good. The team Avengers is one of the best teams. You people know about it already. Okay, so I am starting the human physiology in my batch, so join it ASAP, students. Join it ASAP. Clear? So answer this question, and then let's end the class. That if you remove the fimbriae from the bacterial cell, which of the following would you expect to happen? What should be the answer? What should be the answer? What should be the answer? Be quick. Exactly, the answer is going to be B. If fimbriae they are removed, the bacteria will not adhere or join to the host tissue. Clear, bache? One more question. Mesosomes are the infolding of the cell membrane which? Hanji, which? Quick. See, it's very simple. Are they present in eukaryotic cell? No. So one should not be in our answer. Eliminate these three options and you will get your answer B. Okay. So sometimes elimination method, it helps. Okay. Sometimes elimination method, it helps. So I'll share the PDF in our official Telegram group. Okay. Okay, so you can join it. Again, the link is there in the description box. So now tell me if you guys are having any doubt. Do you have any doubt? Do you have any doubt? Okay, so do let me know in the comment section that you like the class or not, and what are the topics that you want me to take first, right? What is the next, which chapter you want me to start next? Okay, chalo. So take care. And you have to do your homework. You have to tell me the difference in eubacteria and archaebacteria. You can mention it in the comment section. And you have to tell me that 50S and 70S, it is made up of which ribosomal RNA and the proteins. Right, students? Just take care. Bye-bye. Thank you so much.